Genomics and transcriptomics can help to dissect these group of patients. First of all, genomics is the only technique with which you can really look at the NSD2 disruption site. The transcriptomics really helped us to understand what are the genomic drivers and the pathways involved in these kind of patients. And let me add that even more important has been the epigenomics, because this group of patients have a hypermethylated genome due to the NSD2 function overexpression. So I think that these three techniques really helped us to move forward in the understanding the biology of these patients.